All right, so let's go over this plasmolysis, this virtual plasmolysis lab. So what you're going to be doing is you're going to be looking at a virtual representation of the roeo plant in hypotonic, hypertonic solutions. And this first part tells you all about plasmolysis and deplasmolysis. We've talked a lot about the diffusion of water, and these are just different states. Plasmolysis is the shrinkage of the membrane. So anytime the cell is losing fluid, losing water, it's going to shrink. That's going to be the plasmolysis of the cell. And anytime that the cell is swelling, it's a deplasmolysis. Okay? Plasma is what the cell's made of. The inside cytoplasm is sometimes called plasma. And so when it loses water, it's plasmolysis, and when the cell gains water, it's deplasmolysis. Okay, so here's some examples of what you're going to see in the lab today. So here's a hypertonic solution. That means there's a lot of solute in there. And so when there's a lot of solute, it means you have little water concentration outside the cell, and that means you have a higher concentration of water inside the cell. And so what's going to happen is the cell is going to lose water. And so what you can see here is the cell walls kind of stay where they are, but the inside cell membrane shrinks. And the pink stuff just kind of represents all the other solute and things that are inside the cell that can't get through that membrane, okay? And here you can see a deplasmolyzed cell. It's a hypotonic solution. Hypotonic means there's very little solute in the solution, which means there's a higher water concentration outside the cell, and so the water will rush in. And when that happens, the cell will swell, and so the inner plasma membrane kind of pushes the outer walls out and just kind of makes them all puff up. So that's why they all kind of look squished together here. And again, the pink represents all the other things in the membrane that cannot get out. Here are the instructions on that. Make sure you read these because you need to kind of follow them in this way. So I'll give you a quick demonstration of that. Okay, so you can definitely check out these other different tabs. They're worth checking out to help you. But basically, you're going to select your solution. And the solution is NaCl, which is salt. So in this case, we'll try the 0.1% solution. And you can't click start or anything yet. And just so you know, the reset button's kind of hidden down here. And after you're done, you're going to kind of identify what kind of solution that the cell was in to make it behave that way. And so what you have to do is pick up the cell, put it on the stage, under the stage clips. And the other thing you have to do to get this to work is to click on the ocular lens or the eyepiece. And when you do that, the cell is kind of set up. You're going to see the normal cell, and this is the user selected cell. So the user selected cell is going to be the one that you're placing in that solution. And I've already selected it before, so now I'm actually ready to hit start. And you're going to see the timer go. It's going to tell you how long it's going to take for this to happen. So it took 4 minutes, 30 seconds. And as you can see, this is what a normal cell looks like. And it looks like for these conditions that a 0.1% solution was probably a hypotonic solution because water rushed into the cell, which means that this is very little solute compared to the normal conditions inside the cell. So I'm going to select something incorrectly first because I want you to see. So if you select the wrong one, it's going to give you a red X. But when you select the right one, you're going to get a green check. So you know that you selected the right solution. You're going to record your information in the lab, in the data table and whatnot, and then you're going to reset this. Okay, and now you're going to start again. You can start by selecting the different solution. Again, the slide has to go on the stage under the stage clip. You must select the ocular lens, and then you're ready to hit start. So do all of these things. Go through make sure you do drawings for the cells in different types of solutions. Record the time, record the solution that the cell was in, describe what happened to the cell and why. This is where you talk about osmosis and different types of either plasmolysis or deplasmolysis, and you talk about the water movement because of the different solute concentrations. And you have that for all the different solutions. Here's a little chart. You're going to use a table and graph. So the concentration of salt solution, the time it took for the water to diffuse through the membrane, and then you're going to plot that as a graph. You don't have to be exact, but you definitely want to get a general trend here. So, put some numbers in as best you can. I realize this is not a lined graph. I'm more interested with you seeing the trend. So if the line goes up and looks like this, or the line looks like this, or if the line looks like this, that's what I'm more concerned with, is that you understand that as salt concentration increases, what happens to the time it takes for the cells to either plasmolyze or deplasmolyze, right? And then you're going to answer these different conclusion questions. And that's your plasmolysis lab. Have fun with it.